Hi, Lois. Hi.
Sébastien.
I am very happy to have you here and to be able to discuss vision with you. We will also talk about blue light in another episode, but we need to start at the beginning to fully understand this phenomenon, its stakes, and how vision works. And so you will be able to explain to us quite simply, maybe in your own terms, all the studies, your entire background. Maybe you can start by introduce your background a little bit so that people understand.
Yes, thank you, Sébastien. The pleasure is mutual. And indeed, we are going to talk about vision. I have the other time when we did a live session together, I mentioned the ebook I wrote, and the first part of the ebook are the basics. It's a major biological principle that a vision, understanding vision before understanding the impact of artificial blue light on our lives. So understanding how vision works. And another major principle is a physical one, which is what light is, and also what blue light is. And so here, with this video, with this live session, together, we are going to break down vision a little bit, going into some details, but not too many. Too many details, and then try to keep it simple. To also understand why it's important, I think, to understand why and how vision works. What is it? It's still something fascinating how each of our cells aligns for this purpose. Our cells align for many other things besides vision, but vision is really something. Then, well, the eye. The eye is our gateway to the world, and it's already a privilege that we can see. And understanding that also allows us to better grasp our observation of the world. And as a result, what we also do in life, I think.
That's it. Knowledge allows for a better experience. And here, the idea, and what I really like about what you're presenting and proposing, is that, like me, you see the natural principles, understand them, and can exploit them in the best way. But we need a bit of knowledge to precisely understand all these little things. So explain to us, go ahead, where do you want to start? So finally...
Well, so I think we wanted to know how we are able to see. That's it, actually. What makes it, scientifically speaking, what is this phenomenon that allows us to see? So, what makes us see is that we have an entire system in place thanks to the eye that connects to the brain. What makes us see what we see, if there's one message to take away from this entire video, it's that the eye is an extension of the brain. The eye is already the beginning of the brain, particularly the retina, to be more precise. So once we understand that, it's already good. And then we can go into more detail. But understanding that the eye is already the beginning of the brain, it already makes us understand that our entire interaction with the world, our visual interaction with the world, in fact, it's not just a sense. It's not just a sense like touch or anything. It's also already part of the nervous system. And so, mood, anxiety, hormones, everything is also regulated by our perception of light and our perception of shapes and then colors. So here we are going to talk about vision, shapes, colors. What makes us see? So as I told you, it's a system that is set up, the visual system. It starts with anatomy. So yes, you told me to talk about my background. First of all, I'm not an ophthalmologist, but when you're in the 6th year of medical school, you prepare for the 6th year exam, and as a result, you have to cover all the subjects. And I quite liked ophthalmology. Anyway, because actually it's something very concrete and very anatomical. In fact, what the people who gave the ophthalmology lectures said, for example, so the ophthalmology residents, and they said, but ophthalmology is simple. Well, I think actually it's not simple, but they would say it like that. Well, ophthalmology is simple. The reasoning is anatomical, and that's what I like. In ophthalmology, it's that actually the pathology is, well, the whole functioning that we can understand. In ophthalmology, it is done with reasoning based on anatomy, and from the moment we know the anatomy of the eye, and also a good part of the nervous system, we understand why a certain pathology occurs like that, etc. So we will start with anatomy, and actually, because the path of light perception and then perception, finally vision, is done anatomically. So, can I share my screen?
If you try, go ahead. We'll see if it works. Well, I don't have the knowledge. Go ahead. Go ahead. I'll explain a little bit what I have in mind, since I'm here to listen to you as a speaker on this subject. For me, vision, the eye, we have the eye, we have the pupil, we always have this triangle scheme in the end, which comes to reflect on the retina. And then we know that the image is inverted. And finally, the brain puts the image right side up. That's about the little knowledge I have regarding the anatomy of the eye. We talk about other little things, but there you go.
Great. Can you see my screen now? No, I can't see your screen at all.
I think it must be... Hi, we should be able to do it another way. Oh, yes, let me see. You're looking. Now, I see your screen and I'm able to display it. So go ahead, tell me if you... Wait, yes, that's it. There you go. Okay, great. So explain to us. That's great.
Yeah. I'm just looking for a good... There you go. That's not bad. So, let's say there's light coming in from here.
Okay.
The light comes in, and the first anatomical structure that will encounter the light is the cornea. The cornea, in fact, primarily serves to absorb a little bit of the light and to slightly deflect it. But we can move on. We'll keep it simple. Next, there's the pupil. The pupil, in fact, is, like in a camera, a diaphragm. It's the focus. Well, it's not the focus. It's not the focus. The pupil is a hole, actually. The pupil is like a window that allows us to say, okay, I want to see this amount of light, or I want to see not much light. For example, at night the pupil dilates normally, if it works well, so that more light can enter, and thus we can see better. Conversely, if we shine a light, if we shine a light into the eye, then the pupil will constrict, so that, because there are muscles actually here, it constricts to let in less light and prevent us from being too dazzled. Okay? That's the pupil. It's a window, it's a hole. And just behind, so after the pupil, very important, is the lens. So all of this, in fact, is really the light path. Alright, light, therefore, is radiation. Electromagnetic, so it's an electromagnetic signal. And the lens, what it does is mainly two things. Its first well-known function is as a converging lens, meaning the light rays hit the lens, and the lens, its main job is to accommodate, in fact, to allow the light rays to converge, so that there is a single focal point on the retina. This is the retina.
All right, okay.
This is the retina. So, it's a converging lens. It converges the light rays onto the retina. I will talk about the retina right after. And so, this lens is very important. And today we accommodate it. As a result, a lot because we look at things up close a lot, whereas in reality, our eye is still made. Not to make its lens work so hard, and to look more often into the distance and with a more natural lifestyle. But, well, and this lens, it's when it becomes pacified due to too much oxidative stress, and particularly because of age, but also due to traumatic causes or too much oxidative stress. There are plenty of oxidative factors, I'm not going to cover them all. In that case, it's the well-known pathology of cataracts. That's it, okay? So that's the lens. And the other thing it does is block part of the light. It's another kind of filter, additional, which helps block a small portion of blue light and physiological ultraviolet rays. So, there, I have talked to you just about the cornea and the lens. I'll make a small digression about blue light. So, the cornea and the lens block a bit of blue light, and the pupil also allows little or not much light to enter. In children, the pupil is a bit larger and the lens is still developing. Actually transparent, so it lets a lot of light through, which explains why children are more sensitive to blue light, ultraviolet, etc.
All right. Okay.
So then the lens converges the light rays onto the retina. And the retina, as I told you, is the beginning of what?
It's the beginning of. Go ahead then.
Of the brain.
It's the beginning of the brain, indeed.
It's the beginning of the brain. In fact, the retina, well then, tell me if it's too much.
No, I'm listening. I'm very interested. So go ahead. We need to understand, if you will. The people who will listen to us, who will listen to you up to this point, it's because people need to understand. In order to understand what the basic principles are of anatomical functioning of everything that will be electromagnetic and colors, and then to really know what we're doing, whether we equip ourselves with glasses or not, what's happening with blue light, where it comes from, which will be the subject of the second series. So here we're really focused on understanding all of this. It's the imprint. There's no problem.
We're not even talking about light, almost not, minimally, and not even about blue light. So the retina. You need to understand that there are many different layers of cells, that there are neurons inside. And so there is the neurosensory retina and the pigmented retina. The neurosensory retina basically consists of neurons, including the photoreceptors that I will talk about, and the pigmented retina as well, which I will discuss. And so these have two distinct embryological origins. What is embryology? Well, I don't have an exact definition, but it's about how we are formed. There are tissues that have different origins. So the pigmented retina is an epithelium. It's a juxtaposition of cells. It's somewhat like a barrier, if you will. The skin, for example, is a barrier, whereas the neurosensory retina consists of neurons. That's the first major point. I will delve into the neurosensory retina. I will talk about cones and rods.
That's what I was missing. I have this notion in mind of cornea and rod, but explain it to us.
First, I'm just looking for a diagram.
Yes, for sure, cone and rod. It's like the binary decoding we can have on a computer, which is what happens at the back of the eye and ultimately transforms all of this.
That's right. In fact, that's what I wanted to say just before talking about cones and rods. There are layers before the light, before reaching the cones and rods, which are the photoreceptors, and allow us to interpret. The cones and rods allow us to count the photons and somewhat gauge their intensity based on their wavelength. And to say, okay, what I see there is this color, or what I see is roughly this shape, depending on whether it's a cone or a rod. I'll come back to that. But just before that, it's as if the light arrives by passing through many layers of neurons, the bipolar cells, so that it can be noted. There are layers of neurons, and it's as if the light arrives silently to reach the back of the neurosensory retina, to touch the photoreceptors, and it's then that the information is relayed by the photoreceptors. And the neurons to then go into the optical pathways, because I haven't mentioned it yet, but the end of all this is for vision. In the visual cortex, so the visual cortex is in the brain. So
did I make you record? I'm following you. Go ahead. I'm listening. I've watched a lot of documentaries and interesting things, so I can keep up. I'm totally following you on the anatomy. It's very interesting. Go ahead. Continue.
Okay. So here we have the cones and rods. So there are these two types of photoreceptors. This is vision as we have known it since, I believe, the 1800s with Dr. Franz Boll, and then others. After that, there were others. But basically, there are the cones, which are few in number. So the cones are about 6 million per eye, and the rods, which are very numerous. They have very distinct functions. And also, they have a distinct anatomical location. So I will start with the rods. The rods, you should think of cats. Cats have good night vision, and they don't see very precisely. In fact, the rods are numerous, but not very precise. The rods are very sensitive to light, so they allow us to see a little when it's very dark, but they don't allow us to see precisely. So the rods, in fact, they are anatomy. The rods, 120 million per eye, they are mainly in the peripheral retina, meaning they simply line the retina like this. They line almost the entire retina except for the macula. The macula, I will talk about it again. And so these rods, they help us to see in the dark and to perceive shapes. That's what they allow us to do. The rods are therefore very numerous. Now the cones, they are what allow us to see during the day. And that's what allows us to see precisely. It's the cones. There are 3 types of cones that were discovered precisely by Dr. Franz Boll. There are those that are sensitive to wavelengths, mostly blue. Those that are sensitive to wavelengths, mostly green, well, they give a rather green color. And those that are sensitive to mostly red wavelengths. And that, I'm getting into details, but like this, these are primary colors, that's it. And consequently, we derive the vision of all colors. Well, I think these aren't the primary colors, because the primary colors, I believe, are blue, yellow, red, but that's true. But it's a big difference.
It's blue, yellow, green?
It's actually blue, green, red, So, I was going to say, sorry, I'm getting a bit into the details, but like this, there are people who are advanced who can say, okay, that's why. Blue, green, red, actually, it's the presence of these 3 types of cones. This is the basis of the Young-Helmholtz trichromatic theory. You can look it up on Google. And this theory, it's something we can also see a bit in the final year of high school and all. It explains that when these blue cones, green cones, and red cones, which are sensitive to blue, green, and red, are stimulated at equal intensity, that's what makes us see white light. So, that's the trichromatic theory. And so I'm coming back to the cones. The cones, I would like to show them to you. The cones, they are located in the macula.
Okay.
The cones, there are a few of them. There, you can see it well. The cones, they are located in the macula. The macula is really the area where we see precisely. Macula, it can remind us of AMD, age-related macular degeneration.
All right.
I'll talk about it again, but that's where it comes from. In fact, age-related macular degeneration, it's when the macula ages and it doesn't work anymore because the cones degenerate and the retinal pigment epithelium as well. The center of the macula is called the fovea. That's actually how I named my brand at the beginning.
All right.
And so in the fovea, there's even the center of the fovea called the foveola. It's a very central depression, very small at 0.5 millimeter, and yet it's really what allows us to see precisely and to see colors. All right. So it's very important to understand that cones are few in number, very precise, and also fragile. They are fragile. That's why the question we said we were going to ask ourselves is, why is what we're talking about important? One of the main points is that cones are few in number, very fragile. For example, in retinal detachment, so that it's something that can resonate with people. Cones degenerate instantly. And this explains the rapid decrease in visual acuity in retinal detachment. So, there you go, cone rod.
Cone rod, great.
Okay. Next, so now, we are in the neurosensory retina. These cones and rods, what do? As I told you, they count the photons. They say to themselves, okay, for the cones, here, I see more blue, there I see more green. And then they really do a whole sum. It's something that is cyclical. That is to say, in fact, they have discs. And those discs contain photopigments. Rhodopsin for the rods and iodopsin for the cones. And these discs are bleached by the light. And then Their second function is not just counting. They don't just count the photons, they transmit to the brain. So that's really phototransduction. They transmit the information to the brain. They tell the visual cortex. I saw X photons with such intensity. So now it needs to make green. And as a result, we see green, and we see such a shape and everything. And so that's what they do. And now how can they do that? It's also because there's a second part of the retina, which is the pigmented retina. And that part is the retinal pigment epithelium. So, as I told you, an epithelium. Epithelium, it's a barrier. Barrier, it's a juxtaposition of cells. I will, so it's there. In fact, we see it here, you see there, and that you are even pigmented.
Okay.
I would like, there you go. The covered to fight us, and I am just in a place where they really put the there you go, good. Well, there, it's good. We can see clearly. Here we have the neurons we were talking about. We also have cone rods. And here, this is the retinal pigment epithelium. Here, in red, it's the blood, it's the choroid. All right? It's unique to the retina as well, and it's blood. It's important. So why? Because like in many systems, in many organs we have the heart that pumps blood. The blood arrives with oxygen, with nutrients, with lots of good things for the cells. Yeah. And the eye is no exception to that. Well, the retina, the photoreceptors, they also need the nutrients that are brought by the blood. And so there is a barrier for that, somewhat like a filter. It's the retinal pigment epithelium. The right way to understand the retinal pigment epithelium, even if I don't know everything, okay? But regarding the retinal pigment epithelium, it's like the best friend of the photoreceptors because it allows them to live, it allows them to function. For example, I was telling you about the discs that are periodically renewed for the cones and rods so that we can see this visual cycle. Well, that's made possible thanks to the retinal pigment epithelium. Without the retinal pigment epithelium, the photoreceptors do not work. So the retinal pigment epithelium will allow the delivery of nutrients. It takes the nutrients from the blood and gives them to the photoreceptors, but also once the photoreceptors have done their job and have waste, to dispatch, I want to say, the retinal pigment epithelium takes them and then dispatches them into the blood. You see, it's a cycle. And so this retinal pigment epithelium, in fact, there is no other. I found an old one. You still see my screen, yes? Okay. What
is interesting to understand is that ultimately the composition of the blood, the internal water, since it's extremely important to take care of it. And having good blood fluidity also provides benefits for vision. And especially not to make oneself sicker, or in any case, to protect oneself from certain things. So you were talking about oxidative stress. It's really also linked with the connection to the earth, which allows action on the blood. So we really realize that ultimately our two worlds intersect at some point. And that's why the natural physiological understanding of things leads to this knowledge, to having a life experience, a view that is as optimal as possible.
Completely. You really make a perfect transition there. A perfect transition to oxidative stress. We're not really going to cover all of oxidative stress, but that's exactly what I was getting to, without even talking about blue light. But, for example, just the importance of eating well, or having other antioxidant factors, but really eating well, to have quality, antioxidant capacities, and also walking barefoot in nature a little bit. But all of this is because the retinal pigment epithelium consists of cells that are very sensitive to oxidative stress. In fact, so here, this is. Here you recognize the rod with the disc. This is really a diagram I made a long time ago. We are not going to go into all the details. Here we have a photoreceptor, and here we have a cell of the retinal pigment epithelium. The two really work together. The retinal pigment epithelium allows the photoreceptor to function properly. The photoreceptor does its job, as I told you, of phototransduction. It analyzes the photons and everything. Then, a membrane degradation of the outer segment. It's a bit complicated, but just that thanks to light, because oxidation is still a physiological process, normally. Thanks to light, there is an oxidation of this outer segment. And then this outer segment must be degraded by the retinal pigment epithelium. As I told you, it's about the waste that is degraded by the retinal pigment epithelium. But when the degradation is incomplete, because there have been too many oxidizing factors, for example, the person ages, it's normal. We are all heading in that direction. But, or if the person is exposed to too many oxidizing factors, whatever they may be, poor diet, no exposure to natural light, too much exposure to artificial light, there are still many oxidizing factors, especially in today's world, and in that case, the retinal pigment epithelium will accumulate a pigment that marks the aging of cells. I haven't been able to read about any real functions of lipofuscin yet, so it's a pigment called lipofuscin. There must be some functions, but in any case, it is mainly a pigment that accumulates in the cells and is not really friendly to them because it makes them dysfunctional. And moreover, there is a vicious circle where, due to oxidative stress, lipofuscin is created in the retinal pigment epithelium, and the stress, it is stressful, and the stress is stressful, and this lipofuscin itself induces oxidative stress. Oxidative stress, which in turn causes the death of the pigment epithelial cell, and consequently the photoreceptors as well. In fact, this is the mechanism of AMD. It's a major part of the mechanism. There are two types of AMD. AMD is too much oxidation, too many oxidizing factors that cause the photoreceptors to die. There you go. With this accumulation of drusen. This is in dry AMD. Dry. I won't go into too much detail. Basically, too many oxidizing factors, the retinal cells suffer. Okay, there you go. I will come back to this. So, there you go, that's already there. We already understand a large part of vision. Then, because there's no need to go into more detail, it goes to the brain, to the visual cortex, and it's analyzed. And then, there you go, it goes into the optic nerve. There are visual pathways, but I won't go into detail because it's not important unless you're a neurologist.
So I have a question that comes to mind. Thank you. Speaking of the anatomy of the eye, what causes a blind person not to see? That's interesting. What malfunctions in the anatomy? Is it an anatomical dysfunction that causes complete loss of vision? So I think it's primarily an anatomical reasoning.
It all depends on where the lesion is or where the addition is.
There can be several causes.
Well, I don't know them all, but they can be traumatic causes. So someone who had a car accident and has a lesion at the level of the optic chiasm, or I don't know, at the cerebral level as well.
All right. There can be, I thought you break your arm, you know it's broken. And there you could say it's the rods that no longer work, or you could have the retina or the lens. So it could be any of those you've mentioned, which can malfunction and jeopardize everything else.
Exactly. And there are really many different types of blindness, if I may say so. There are those who are born blind, and then there are those who they have had AMD and consequently have a real degeneration of their photoreceptors. While everything else works well, so to speak, but at the level of the retina, it doesn't work. That's AMD. Cataracts too. Cataracts eventually lead to a decrease in visual acuity after a certain period. And cataracts, as I was telling you, involve the lens. So, In fact, there are many, many, many causes. I don't want to get into it, but there are many causes for being blind. In the end, what is the best answer? It's just a dysfunction, which can be due to an injury, but it's a dysfunction or a loss of everything, of one of the things I just mentioned. So I even think of a cornea, which, there you go, imagine a cornea, that no longer allows light to be absorbed and is completely opaque. I don't know if that exists, but it would make people blind. There you go.
Great. Thank you. It's really very comprehensive. If there are any questions in the comments under the video, don't hesitate. You can answer them. And then we'll meet again in a future video to talk specifically about blue light, about light. What is the phenomenon that occurs? What disturbs us?
In a future video, it will be about the physical principle of what light is. It's a wave, so definition of a wave, definition of a wavelength. I'll need to review a little bit. Thank you. Definition of a wavelength, and so there you really have it. Definition of that, and then how does the perception of light occur? Because here we talked about vision, certainly. We see light, we see colors, we see shapes, but then how does the perception of light occur, and why is it important? And actually why it's almost, I want to say, I'm taking a stance, but I think it's almost more important than seeing colors, because perception, actually, already from an evolutionary standpoint, if we have eye sockets, if we have eyes that fit into the sockets, it's primarily to see light more than to see colors and shapes. I'll say it again. It's to see more of the light, just a light signal, rather than saying, okay, it's blue, it's green, it's square, it's orange, there you go. Because the perception of light, so that's the next video, but the perception of light, it's the first thing that sets, that regulates, that synchronizes. Our base, really, our foundation, which is our biological clock. We are made to live according to the Earth's rotation around the sun. Day, night, and that. It happens primarily thanks to light. There are other pathways, but the very first is light.
You had proposed a question to me in preparation. Why is it important to understand the phenomenon of vision? And you had mentioned the spiritual aspect.
In fact, I've already briefly talked about it, but in relation to the experience of life, actually, that's it. It's the fact of observing the world, the fact of seeing the world well, the fact of seeing it clearly and not having dirty lenses, and not when wearing glasses. The fact of seeing clearly, and then also understanding how fragile our physiology can be. Also, if we don't take care of it in the long term, if we don't understand that and if we neglect it, I think, I'm sorry to use negative terms like that, but it really leads to disaster. And on the contrary, if we open our eyes and marvel at the beauty of the world, and that, in any case, that's my situation. I marvel both at the beauty of the world and also at thinking, but it's crazy that all these cells coordinate with each other to do this. And for hundreds of thousands of years, it's only since the years, as I told you, the 1800s that we understand photoreceptors, etc. But yet, 100,000 years ago, Homo sapiens, they had the cones, they had the rods, they had the same retina. And as a result, neglecting this is neglecting a part of our nature. Forgetting this is forgetting a part of our history and our nature. So, what's the point of knowing this? Simply to be able to take better care of it.
That's it. Knowledge allows for a better experience. Listen, on that beautiful note, thank you, Lois, and I'll see you very soon then.
Thank you, Sebastian. Thanks to the partnership with Plus Chow.